What's up, folks? So here we are doing some Unit 3 review. I got Unit 3 pulled up here. All the things from Unit 3. Remember, Unit 3 goes all the way back to enzymes. You probably done forgot. And we got cellular respiration and cellular and photosynthesis. All the fun things. This looks like maybe this is an old one, maybe? There's some old pictures. I can't tell. Yeah, here we go. This is the new stuff. It's all the same. So my plans are to go over some con questions with you guys, and if you have specific content questions, I can do that too as well. And yeah, now Unit 3 is a lot shorter. So all right, so let me pull up some con questions here. I'm just going to start with enzyme stuff. Sort of go from there. Yeah, it's time for a quiz. And like I normally do, I'm just going to copy paste them into OneNote here and not remember how to use OneNote. There we go. Okay, so here's question number 1. Cyanide is a toxin that can affect cellular respiration. It functions by binding to an allosteric site on the enzyme cytochrome C oxidase, disrupting the electron transport chain. Cyanide is able to bind to cytochrome C oxidase, whether or not the enzyme is bound to a substrate. Okay, so it looks like this is pretty easy, so I'm not going to even go through, but let's go through the question anyway to help some of you all. I know you like to hear me kind of work through the question. So as soon as cellular respiration is going to trigger certain things, right? And to an allosteric site. Now I know that I'm looking at an enzyme. I'm looking at an enzyme, and I'm looking at this cyanide that changes the shape of that enzyme by binding to a site that is not the active site. And then I'm given some more information about electron transport chain there. And so let's go. Which of the following best describes cyanide? It is a non-competitive inhibitor, right? Because a non-competitive inhibitor does not bind to the active site. Cofactor, we didn't really talk about those. Allosteric activators, probably not a thing. Really, it's between C and D. And since allosteric site is not the active site, C is the correct answer here. We got to get the little bling sound. I just realized that my sound is it's worth it. Okay. Next question. Ooh, this one's got a graph. Stop me if you have questions. I can't even really see if anybody's in the chat or not. So, I don't know what's going on. I'm scared to push buttons because I don't want to turn my stream off. I'm still pretty unfamiliar with this particular type of software. So, all right. So here's a new one. Consider a reaction in which reactants X and Y combine to form Z. The diagram below compares the reaction coordinates for the catalyzed and uncatalyzed pathways of this reaction. Which the following about letter C is true. Okay, so we've seen this graph before, right? We have energy on the y-axis. We have reaction coordinate on the x-axis. Sometimes you'll see it called reaction pathway. It doesn't matter. You have the products on the left, the reactants on the right. X and Y turn into Z. And we remember that those humps B and C represent the amount of energy that's necessary to get that reaction to start, otherwise known as the activation energy. This is a vocab question. Let's go find that one in A, B, C, D. Letter C represents the activation energy of the catalyzed pathway. Oh, yeah, it's making us decide between C and B. Oh, yeah. Well, the catalyzed pathway is going to have a lower activation energy, and since C is higher, A is incorrect. Letter C represents the activation energy of the uncatalyzed pathway. B is correct. I don't know what these other two are other than being wrong. They are wrong. All right. Moving on to the next question. Although organisms are highly ordered systems that may appear to contradict the second law of thermodynamics, they don't actually violate this law. Which of the following statements best provides the best explanation for why living things do not violate the second law of thermodynamics? We remember the second law of thermodynamics has to do with every energy exchange represents a loss of heat, and that's because entropy increases in systems, right? There's no such thing as a system that does not increase in entropy with every energy exchange. And so living things demonstrate this in a number of ways. We're just going to go down and find in ABCD something that aligns with what we know about second law of thermodynamics. Let's look at it. A, the amount of energy in the universe remains the same when chemical energy is converted to kinetic energy in a cell. This is kind of a tricky one because it has, first of all, it's the first law. Energy is not created or destroyed. It's transformed or transferred. This is true, but it doesn't have to do with the second law of thermodynamics. Organisms require a constant input of energy to maintain order, and some of that energy is released into the environment as heat. This is exactly what we're looking for. I can go with B with some amount of confidence, but we're going to look at C and D just to make sure. All processes carried out in the cell result in the release of heat into the environment, which increases the total amount of energy. That is not true. Energy is not created or destroyed. And then let's look at D. Order is maintained by biological processes that occur in an organism, and these processes decrease the total amount of entropy. They do not. Entropy only ever increases. B is the correct answer. Good question. Good question. Chat's been fairly silent. Makes me wonder if I'm doing something wrong, but I don't think I am. Everybody's just listening. Okay, here we go. Several scientists are studying the effect of initial substrate concentration on the activity of an enzyme. They perform a series of enzyme-controlled reactions, keeping the concentration of enzyme constant, but increasing the initial concentration of the substrate of each trial. The graph below shows the results. Based on the data above, which the following best describes what happens to the rate of product formation at higher initial substrate concentrations. Okay, so they keep the amount of enzyme constant, but they increase the amount of substrate, and then you can kind of see what happens. Product formation increases and then levels off as substrate amount increases. So initially, it increases quite a bit and then slows and then levels. And so what are we asked to look for? Appreciate that. Thank you very much. Appreciate that vote of confidence. So what are we being asked to look for here based on the data above? Which of the following best describes what happens to the rate of product formation at higher initial substrate concentrations? Let's just go look at the answers. This one's kind of tricksy. Higher initial. I'm thinking like toward the upper end of things, right? The rate of product formation levels off because no substrate is available to bind with an enzyme. No, that's not how that works. The rate means that product is still being made, right? There's just a certain amount of enzyme. Because the enzyme is static, the rate is going to level off. That is not it. The rate of product formation levels off because nearly all the enzyme is bound to substrate. I don't like that one because of the language, bound to substrate. Let's look at C. The rate of product formation decreases, it doesn't. The rate of product decreases, it doesn't. So we're stuck between A and B. We're going to go with B here because all the enzyme is taken up and only doesn't matter how much more substrate you put in, the enzyme is taken up and can only react so fast. We're going to go with B here. I don't like the wording in this question. But you know what? We don't have to like the wording in the question. So again, let's talk about our logic there because that one was a little more tricky. So we know that C and D were wrong just because the graph show us that they're wrong, right? And A and B, so that's what we're stuck with. The rate of product formation levels off because no substrate is available to bind. That is not true, right? Because we can see that substrate is being put in more and more, and so that is not true. I'm going to text someone in my family to tell them to put our stupid cat outside. All right, and so B is the correct answer. Moving on. All right, let's look at this question. Acetylcholine, ACH, is an important signaling molecule in the nervous system. After it transmits a signal, ACH is broken down by the enzyme acetylcholinesterase in a reaction known as ACH hydrolysis. Which of the following best describes the effect of acetylcholinesterase has on the hydrolysis of ACH? Big words, but symbols to make it a little easier. So hydrolysis is a reaction that breaks down something, right? Adds water to break down something. What is the enzyme doing here? It's just catalyzing that reaction, slowing the activation energy. Don't make this one more difficult than it is, right? Which I was just trying to do, if you can believe that. The enzyme it's all it does. It's catalyzing a reaction. Hydrolysis is a type of reaction. Catalysis, I don't even know how to say it. Catalyzing a reaction lowers the activation energy, making the reaction happen faster. Let's go find that ABCD. Increases, A and B are both wrong. Decreases activation energy, increasing the rate of reaction. C is the correct answer. Good. Hey, we made 100, and we leveled up some skills. Congrats, everybody. Job well done. Okay. So that was enzymes. Moving on to We got photosynthesis. There's another quiz. Let's do this one. So this next quiz is going to be photosynthesis, cellular respiration. It doesn't look like Khan has upgraded its sections. Content's still the same. College Board just kind of changed its sections up a little bit. And so if you're doing Khan questions, you'll see a section called fitness. That used to be 3.7. It's not a thing anymore. It's not wrong or bad. It's just not a section. Okay. This is quiz 2 concerning all things photosynthesis and cellular respiration. Wow, this question is the longest question in Khan history. I don't know if I can fit it in my screenshot. Let's see. They should like flatten some of these questions out. They should accommodate my wishes. It's just not how things work. Okay, so this is a pretty big question. Let's look at it. Phospholipase proteins are a major component of snake venoms. When an animal is bitten by a venomous snake, these enzymes, phospholipase proteins, these enzymes catalyze the breakdown of phospholipids and can disrupt cell membranes, leading to muscle damage and other effects. Snake venoms often contain multiple types of proteins, each with a slightly different amino acid sequence. The pit viper, Photobothrops flavoridis, contains multiple enzymes, including BP1 and BP2. The amino acid sequences BP1 and BP2 differ by only one amino acid. A group of scientists tested the enzymatic activity of these two things, and you can see the shown there. Based on the information provided above, which of the following statements is most likely true? Okay, so sorry, I've got things going on in my house, and I was trying to read that question and also understand what it was asking at the same time. Two kinds of enzymes, and these enzymes break down phospholipids. Okay, and then enzyme activity is a higher rate with BP2. Okay, as you increase. Okay, so based on the information provided above, which of the following statements is true? With the exception of that, a snake's venom often contains a single type of enzyme, so that its effect on prey will be physiologically targeted. I don't know why I would need to believe that, based on what we see here. That venom likely has a narrower range of physiological effects. I don't know why I would believe that either. The active site of BP1 is slightly different than that of BP2, so the two enzymes function differently at most concentrations. Again, it's so funny. You could probably just read the bolded question and then the answers and not even read all this about venoms and Photobothrops and all this stuff. And this is just a true sentence. Let's look at D. Amino acid difference between BP1 and BP2 causes BP1 to have a higher activity level than BP2 at medium and high concentrations. I don't know why D would be true. This is a weird question. I like C. It may be wrong though. No, it was right. Yeah. So let's go back and look at this one. I think this is and we've seen a few of these. We do these in class. If you're not in my class, we do a lot of these questions every time we lecture. And some of this stuff, like based on the information provided above, right, which is a graph in this passage, which of the following is most likely true? Everything but C is sort of making something up, right? It's like, it's making a thing up that wasn't there. And you might even look at D and be like, the amino acid difference between them causes is the word here, to have a higher activity. It does have a higher activity. That's true, right? And Actually, it just doesn't have a higher activity. So but let's just say it was the reverse, right? Let's just say that BP1 had a higher activity. This graph doesn't suggest that it's the amino acid difference that makes that cause, right? And so you have to read the answers carefully. C is true regardless of what system that we're put in. And so that was why it was the right answer. Okay. Wait. No, I don't. Okay. Khan has all these ads and stuff that come up. And I don't know what kind of question that was. It really didn't fit in any of the... It was probably that fitness thing. It was fun. It's not a bad question, though. Golly, so much bigger. Okay. In mitochondria, the rate of ATP synthesis during oxidative phosphorylation is tightly coupled to the movement of electrons through the electron transport chain. When ADP levels rise and the demand of ATP synthesis increases, electron flow also increases. Based on the information above, which of the following best describes the effect of rising ADP levels in describes an effect of them? So if ADP is rising, that means ATP has been spent, right? When ADP rises, the demand of ATP synthesis increases, electron flow increases. If electron flow increases, it's because more electrons have been dropped off by electron carriers, right? And so with increased demand for ATP, you're going to have an increase of glucose being broken down, and all of those steps are going to increase. And so let's see if we can find that ABCD. NADH, FADH2 will be oxidized less rapidly. Not the case. It'll be more rapidly, right? Oxygen consumption will increase. Yes, it would. I like B. If it was me, I'd probably mark it, but we're gonna read the rest of them. The flow of protons through ATP synthase will decrease. Nope, it will increase because it's going to be making more ATP. Fewer protons will be pumped. Nope, more will be pumped. B is the correct answer. Oxygen is the final electron acceptor of cellular respiration, and if more electrons are flowing through, more oxygen is being consumed. Next question. It's another long one. It's not as long as that one, though. That one was the longest question in Khan history. Let me go up here. All right. A group of students are conducting an experiment on Elodia canadensis, species of aquatic plant. In order to test the effect of light intensity on the rate of photosynthesis, they place Elodia canadensis plant into an aquarium tank and place each tank at a different distance from a light source. After a few minutes, this is the experiment I need to do. I need to get some Elodia instead of like Kroger spinach. After a few minutes, oxygen bubbles begin to form in each of the tanks. Students count the number of bubbles released during a 5-minute period for each tank. The results of their experiments are shown below. Plant ABCD, the distance of the light source from the plant, 20, 40, 60, 80, and then more bubbles. You get the idea. Which of the following conclusions best supports the results of the experiment? Okay, so let's look. This is one of those data tables, and they do this all the time, and you have to be careful because the distance from the light source is kind of inverse, right? The smaller the number, the more intense the light, because we are so like trained just, I don't know, through life that more equals better, right? That when we see 20, 40, 80, 60, 80, we're going to think 80 most intense, 20 least intense. But in fact, this is the opposite, because 20 centimeters away is the most intense light, and we would expect that most intense light to have the most bubbles, and that's exactly what we see. And so let's go and see if we can find that ABCD. As light intensity decreases, the production of photosynthetic products remain constant. It doesn't. I don't know. Let's see. As the light intensity increases, the production of photosynthetic decreases. No. No, and D is the correct answer. Polyester sweatpants. You nailed it. Good. Oh boy. I mentioned that we had the longest question ever, but I don't know. We'll have to put them side by side and see. This one's pretty long too. Oh, wow. Okay. I stand corrected. This is the new longest champion. All right. It's kind of blurry a little bit, but we'll live. Photosynthetic organisms use pigment molecules to absorb light energy. There are many types of pigments. Each type can absorb light in a certain range of wavelengths. Photosynthetic organisms often produce more than one pigment. And then we have a bunch of stuff. Many photosynthetic organisms live in aquatic environments where light quality changes with depth. And then you see that there too. Wow, I've never seen this graph before. That's first. Okay. All right. Based on the information provided, which of the following photosynthetic organisms can best absorb wavelengths of light present at depths of 25 and 100 meters? Okay, 25 is here. It looks like all visible. So 25 and 100, and it looks like the answers are providing 2. At 25, the whole spectrum is available. At 100, green is pretty much the only thing that is left. And so a plant that is absorbing green is this FICO, that one, right? And so, okay, let's talk about these graphs a little bit, because these are pretty important graphs. What you're seeing here is absorption on the y-axis, and then this is just a spectrum. And so when you see a line up here, that means that light is being absorbed. All the other lights are being reflected. And so when light gets down to 100, we see that green is the only light that gets to 100. The only plant that can use green light is this one, right? As far as the other ones, this is a full, at 25, everything is on the spectrum. And so which one does it best at that? Probably chlorophyll A, because chlorophyll A is able to get a lot of this and a lot of that. And so let's find chlorophyll A and FICO, that one. I see FICO. Oh, wow. I need to be able to spell it right. It's not cyanin, it is earthrin right here. Notice we didn't have to decide between chlorophyll A and chlorophyll B. Chlorophyll A is on 3 of them. This is not correct, and so we just had to find, really, as soon as we found the purple one, we had the correct answer. C is the correct answer. That was a little tricky. Not that it was that hard. It was just a lot to look at. This is another one about cyanide. Surely they're not giving me the same question, are they? I guess I can't be a beggar. Our beggars can't be choosers. No. Okay. Cyanide poisoning is a type of poisoning caused by exposure to certain cyanide-containing compounds such as hydrogen cyanide and cyanide salts. Inhalation or consumption of these compounds can cause histotoxic hypoxia, a condition in which cells no longer are able to take up or utilize oxygen during cellular respiration. As a result, ATP production during cellular respiration is significantly reduced. Based on the information above, which of the following is the most likely explanation for how cyanide causes histotoxic hypoxia? Okay, so we know that cellular respiration is a 3-step process, right? Glycolysis, Krebs, and ETC. and that Krebs and ETC both require the presence of oxygen in order for them to occur. Krebs is primarily making electron carriers, and those electron carriers are going to ETC, and that is where the bulk of the ATP is made. There is a small amount of ATP that is made in glycolysis, and glycolysis does not need oxygen. So based on what we know, ATP production is drastically reduced during this lack of oxygen. Let's go find that in ABCD. Cyanide inhibits the transfer of electrons to the final acceptor in the electron transport chain. I'm going with A. You know what? I don't even want to read the rest of them. Not even reading them. Not even reading them. Apparently I got a special ding right there. Let's go look at the other ones, though, and see why they're wrong. Cyanide inhibits the enzymatic breakdown of glucose. I mean, that would definitely cause a problem, but nothing in the prompt tells us that, right? And so we're not going to overthink that. Well, no, we're not going to overthink that. We're not. Cyanide inhibits the transport of pyruvate across the mitochondrial membrane. Again, right, we are told that this is a condition in which cells no longer utilize oxygen. And so pyruvate going across the membrane is not going to be a part of that. And inhibits the reduction of NAD plus and FAD during the Krebs cycle. Again, not important. A is good. Is it A because of ATP synthase in the ETC? Yeah, it's A because, yes, it is, but ATP synthase is only happening because the electron carriers are able to drop off their electrons. And why are they able to drop off their electrons? Because oxygen is present. If oxygen is not present, then they're unable to do that. And so the presence of oxygen allows for those electrons to be dropped off because oxygen takes those electrons in, right? It's sort of a dumping ground for those electrons, and then oxygen combines with those electrons and makes water. Without that NADH, FADH2, they're sort of like left with nothing to do, and that's when fermentation occurs. So yeah, good question. Hey, up next is the unit test. I think we've got this. It's 10 questions. Pancreatic amylase is a digestive enzyme found in the small intestine. It helps break down large starch molecules later in the digestive process. Pancreatic amylase is unable to break down lipids. Which of the following best explains why pancreatic amylase is able to break down starches but not lipids? It's because starches and lipids are shaped different. The active site of pancreatic amylase is shaped specifically for starch and not for lipids. Let's go find that in ABCD. Lipids are too hydrophobic. Nope. Lipids are too small. Don't even know what that means. Enzyme binds only to specific substrates. C is the correct answer. Now, I already know some of you, because I know you pretty well, that you're going to look at B and C and you're going to think they're similar. They're not. B is too specific, right? We don't know enough for B to be the correct answer. C is right no matter how you slice it. But in order for B to be correct, other things also have to be true that we don't know about. And so they are not the same, right? B is too specific. C is general enough, and it's also true. B may be correct. We just don't know that from this. We know that C is correct, right? And so that's why we go with C. All right, let's look at this one. In many animals, Chemicals from the environment can be detected by olfaction, or sense of smell. The ability to detect odorants can be important for finding food mates, establish territory, avoiding danger. Odorants are organized by a group of similar proteins called odorant receptors. These receptors recognize one or more specific chemicals from the environment and relay information to the brain. The brain then interprets the combination of stimulated receptors as a certain odor. Animals typically have a large number of ORs encoded in their genomes, as demonstrated in the following table. And there's some stuff. Given the information, mice can smell that much? Really? Given the information provided by the table, which the following predictions are most likely? Well, my guess is that mice and dogs are going to need their sense of smell more than the other animals. Let's look. Dogs are less likely than chimpanzees to be able to detect. No, they're not. Humans have the ability to detect greater number. They just don't. Chimpanzees are more likely to than mice. They're not. D is the correct answer, even if we don't read it, because A, B, and C are wrong. But let's go read D and make sure. Mice have the ability to detect greater. This is a third grade question. Moving on. Now, I say it's a third grade question. Anyone could misread it or whatever. But it's a whole lot of words for you to just compare numbers, which is just what the AP exam does sometimes. Some of the questions are going to be like, I can't believe this is so easy. Some of you guys are actually tripped up by that. All right. Group biology students want to study the effect of pH on enzyme activity. They measure the activity of enzymes A and B in various pHs, and they record the data below. Solution of enzyme A is a pH of 8 based on the graph. Which pH change would likely result in higher enzyme activity? Oh, okay. I'm seeing that it confused me reading that the solution has a pH of 8 and then I see the 6. So what we're seeing here is that the highest activity for A is 6 and the highest for B is 10. Those are the optimal pHs. The solution needs to be decreased to 6 in order to have the higher enzyme activity. Let's go find that ABCD, and there it is. It's C. That was not hard. This is about optimal pHs. Not a hard question. Which of the following best indicates that light reactions of photosynthesis have completed and that the Calvin cycle has begun? Oh, interesting. Let's see if we can formulate our answers before we look at them. Which of the following best indicates that the light reactions have completed, Calvin cycle has begun? NADH and ATP have been created. Oxygen's being given off. There's a number of things that can happen here. CO2 is being fixed. Glucose is being made. Let's go find anything like that. Electrons are transported across the ETC on the thylakoid membrane. That would be during light reactions. So that since they've completed, A is incorrect. ATP and NADPH accumulate in the stroma. There you go. ATP and NADPH are things that are made in the light reaction. They've accumulated in the stroma as the Calvin cycle has begun. I'm going to go with B, but let's look at the rest of them. Electrochemical gradient, again, that's light dependent. High concentration of carbs is found in the thylakoid lumen. That would be after Calvin cycle because glucose is being made at the close of Calvin cycle. B is the correct answer. Ooh, it's a long one. All right. ATP synthase is a protein complex that is highly conserved across eukaryotes. Protein complex helps generate ATP, which cell uses as a source of energy. Hold on 1 second. The ATP synthase complex is made up of multiple protein subunits that are required for its function. One of these subunits, ATP sync C, is encoded by the ATP sync gene. A group of scientists studied the phenotypes associated with the two different ATP sync mutations in fruit flies. The scientists found that all flies of mutation 1 died as larvae. Most of flies with mutation 2 died soon after pupal stage, and some with mutation 2 survived into adulthood. As part of the study, scientists used a protein gel electrophoresis. This is kind of borrowing from like Unit 6 a little bit. To quantify the amount of this protein produced by mutant flies. All right. Okay. So this question is probably a little bit too Unit 6 for us, but we're going to look at it anyway. So there's the control. All of them are found there. And in this protein with mutation 1 doesn't have this protein, which is an important part of the complex, which the following claims is best supported by the study above. In flies with few functional ATP synthase complexes compared to the wild type, stored ATP provides enough. Nope, ATP is not stored. Flies with higher than wild type ATP levels are more likely to die. Flies with mutation 2 have fewer functional ATP synthase complexes. The level of ATP in mutant flies is correlated with the severity of the phenotype seen in those flies. I'm going with D. This question's weird, and I was having to think with my cat meowing in the background. You're not going to see a question like that on this test. That was just a weird question. You got it right, though. God, this question is like 1,000 times shorter. Dr. Kumar has identified a new enzyme. Its sequence analysis suggests that it catalyzes the transfer of a phosphate group from one ATP to another substrate and vice versa. What should Dr. Kumar name his enzyme? What the crap? This is a weird question. I don't like this one. Yeah, this is like, it feels like a Unit 4 too, a little bit. No, I think it's a kinase. We're going to ignore this. This question's not helpful. Well, it is a kinase, but you don't have to know that. This is the question about the pit vipers again. It's going to count it as incorrect. Oh well, missed our perfect streak. Guess I'll have this. Won't sleep as good tonight. It's too bad. All right. That enzyme, I'm not even going to attempt Tyalin. I don't know, isn't. The cat apparently is sneaking in multiple times. Not exactly sure what's going on. Maybe I'm going insane. Is a digestive enzyme found in saliva. It breaks down, or it helps break down large starch molecules early in the digestive process. That is unable to break down proteins. This is similar to the one we had earlier. Enzymes are specific to their substrates. C is the correct answer. There's a lot of enzyme questions on this. There was a lot of photosynthesis and stuff too. Wow, this is a simple one. Actually, no, I did not. Which of the following statements is true? Okay. Living cells can violate. No, they can't. Cell processes that decrease entropy within a living system can occur without the input of energy. Incorrect. The net effect of endothermic reactions of an organism is the release of heat into the environment. Is an overall decrease in the... No, it's not. D is the correct answer. I don't even know what it is, but let's look at it. The organization seen in living cells, tissues, and organs is maintained by a constant input of energy. That is correct. That one, they didn't even try on the wrong answers on that one. And this is the last one. We're going to look at it just so you guys can get reps, but it's going to be a really easy one. I mentioned that a lot in class. I mentioned that with my freshmen and with my AP students. Getting reps, seeing these things over and over again. The reason that I'm able to do this so quickly and easily because I've seen it 1,000 times. Obviously, you can't see it 1,000 times between now and the test, but seeing it 30 times is reasonable and is helpful. So just reps is good. It's like any skill. Group of biology students wanted to study the effect of pH on enzyme activity, and you see that based on the graph. What is the approximate optimal pH for enzyme B? It's going to be 10. B is the correct answer. Hey, we did good. Up next is Unit 4. We're going to move along. Looks like I've been streaming for about 45 minutes, which is good. I haven't had any specific questions in the chat. Went over a ton of con questions, as you can see. And I feel pretty confident. I don't know if you do, but I feel like you guys are going to do well. I'll wait a few seconds for any other questions, and then I will sign off for the evening because I am starting to... definitely starting to wane. I'm hearing a cat constantly. So, all right. You know where I'll be first thing in the morning. If you have questions, come and see me. It's been fun. Oh, yeah, I can. Could you go over ETC a little bit? Yeah, let's do it. Okay, so here's the electron transport chain. I'm assuming you're talking about cellular respiration. Yeah, that picture will be fine. Purpose of the ETC is to use electron carriers like NADH and FADH2, which you don't see on this particular picture, to make ATP. How do they do that? Well, the energy from NADH is going to power these proton pumps right here, right? And they're going to fill the inner membrane space, which is the space between the two membranes, with protons, creating a concentration gradient. Those electrons are going to move through these pumps, and they're going to turn each one of these pumps on. 1, 2, 3, 1, 2, 3 protons. And by the time those electrons get to the end, zoomed in a little too much, my computer is struggling. By the time those electrons get to the end, oxygen is there to take them in. So you have O2, you have these two electrons, they're going to combine with some protons, and you're going to get water, right? Oxygen is the final electron acceptor. This cannot happen unless oxygen is there. So that concentration is built up, and because of that, ATP synthase is going to facilitate diffusion of those protons back into the matrix. And when it does that, it causes ATP synthase to sort of spin, right, for lack of a better term, and that causes ADP to be phosphorylated to ATP. You sometimes see this called oxidative phosphorylation. This idea of a concentration gradient is called chemiosmosis, which is what you see right here. This idea of the gradient being built up and ATP being made on ATP synthase here, or ADP being phosphorylated into ATP. And that's pretty much it. But this word, chemiosmosis, associated with that process, oxidative phosphorylation, associated with that as well. Good question. Good question. All right. Well, I'm going to sign off for the evening. I'll be in my room in the morning if you need me. Good luck with the rest of your study.